This is Bio 181, Chapter 17 on biotechnology. And we're going to talk a little bit about, about some different types of biotechnology. DNA fragments, so they're cut up by restriction enzymes. So these are sequences of DNA. They've got a specific sequence on it. A restriction enzyme comes in, locates that sequence, cuts it, and you've got little pieces, right? And it might cut out, might have a different restriction enzyme here. It's going to cut out a different section. And then you've isolated a section of DNA, which might be a gene of interest. So something that's going to code for a protein that you want to make. So if you say take a bacterial chromosome, so here's my bacteria. Bacteria cells, they're prokaryotic, right? Prokaryotic. And a lot of times genetic recombination is going to use bacterial cells. They're single cell. So they're very simple compared to eukaryotic cells, cells that have a nucleus. So these cells, no nucleus, they have DNA in them. And the DNA is in a single large main or primary chromosome. So this is the DNA. If I was to look at this, right, it would be a double helix. So this is the DNA in the bacterial cell. So here's my chromosome. So if we isolate a gene, say we isolate a gene and this gene is going to allow plants to grow faster, right? So it's going to allow plants to grow faster. Speeds up their, ability to take up some really important mineral. So how do we get that into other plants? So what you often do is, even if it's from plant to plant, you're going to take it out of your original organism, right? You're going to use your restriction enzymes, these little guys, these little Pac-Man guys, to target and find the gene that you want. You're going to cut it out, and then you're going to put it into a bacterial chromosome. So the bacterial cell, you can take it, put it maybe in some heat or some different chemicals, and it will make the membrane a little bit permeable in solution. So the cell membrane, and the gene will actually, if it's put in there in high concentration, will migrate into the bacterial cell and may even integrate and be added into that original DNA. Okay. So when that happens, then if the bacteria divides, it will, for each of the daughter cells, copy that recombined chromosome. And so that gene is going to be copied, and bacterial cells can replicate very quickly. So they'll have exponential growth. And so pretty quickly, you're going to have all of these bacterial cells, and they all have replicated this specific genome with the new gene in it. Okay? And then you can take that and all of these cells, so all of these cells have it, right? And you can take that and you can use that to then later add it into a plant cell. You can put these bacterial cells next to plant cells. There's different mechanisms you can use for that. Transgenic organisms, so that's related. Those are organisms that are created by incorporating DNA from one species into another. So say I've got a corn plant here, right? This is a very common example that I'll talk about later. So here's corn, right? And I want to add in, corn is actually modified. It's one of the most common crops in the United States, corn and soy. So it's been modified a lot. It's heavily modified. There's tons of different genes that have been added to different varieties of corn, and a lot of them are bacterial. So, for example, there's one called BT, and it's from a bacteria, Bacillus thuringiensis. It's a bacteria that is a single-celled organism, and it creates a toxic compound. So it makes a toxin that kills insects, okay, herbivores. So this is really popular. It's a popular way to modify crops because a big risk when you grow a crop is that it's going to be eaten by insects. And so if somehow you can get this a gene from an organism, a bacteria, you take a little bit of the gene out and you transfer it into the corn. It's incorporated into the genome in the cell. So here's like, well, here, let me just blow it over here. So here's a big corn cell, right? So here's a corn cell. In the corn cell, you've got a nucleus. And remember that we have in eukaryotic cells in the nuclear envelope, we've got linear chromosomes. So you've got a whole bunch of chromosomes in here in the nucleus. Okay? And you modify the organism. So now it has, in addition, whoops, this bacterial gene. So I'm just putting in right here, that's going to make that toxin. Okay. So anytime we have two different organisms, a bacteria, a plant, modified, put together, that's called a transgenic organism. So two different species. Another concept that's common in biotechnology is modifying organisms and creating knockout organisms or knock-in organisms. So this would be an organism. So you've got some mice here, right? And you have the normal mice, and then you think, well, I don't know what this gene does. So there's a specific gene of interest. So you've got the mouse DNA, right? And you've got a gene, and you're like, huh, I don't know what that gene does. So you take the DNA, you cut out, right? So you remove, you use, say, a restriction enzyme again to go in and cut out a specific piece, remove it, reconnect the DNA. So it's just less this one gene. And then you use that DNA to create mice, watch the mice grow. And then you say, notice that, okay, without that gene, maybe the mice change color, right? That would be a very simple one. But so it lets you know what that gene actually does. And that's used quite a bit in laboratory settings to investigate the function of a specific gene. So you can take it out and see what effect it has from removing it, if the organism is still capable of living, how it's affected. You can also add in a different gene that would be a knock-in and see the consequence of that. Okay, so I already talked about this. The BT crops are resistant to some insect pests. So that was the example of the bacteria that had the BT gene added to it that was a toxin that conferred resistance to herbivory. So that means if the insects eat it, they die. Another example of a transgenic organism is golden rice. So this has benefited a lot of individuals. It's so generally rice doesn't have too many nutrients in it, especially in certain regions like in India. Traditionally, the rice or often gosh, trying to draw black. So often the rice, okay, would be pretty devoid of nutrition. So there's a grain of rice, but people have access to it in large quantities. It's very affordable. And so they modified this species of rice to contain beta carotene, which is not normally found in rice. So beta carotene is the precursor for vitamin A. And a lot of individuals in countries where they had nutrient deficiencies were running into issues like with their vision because they had a vitamin A deficiency. And so by adding in this gene from maize, which is corn, okay, so they used a gene from corn and a gene from soil bacteria. So they took genes from two different organisms. Bacterium is just singular. So we added into the DNA here, we added, so this is my rice, right? Now I'm adding in DNA from corn, and I'm adding in DNA from bacteria, and the goal of this bacteria is together, okay? It's going to allow for the new corn to produce beta carotene. And so individuals that eat this newly grown type of rice will now have vitamin A without taking vitamins as supplements. So it's one of the positive impacts of genetic modification and transgenic organisms. All right, that's it.